In this video, we're going to look at the first step of protein synthesis and look at how DNA, RNA, and proteins are related. So remember, as a review, DNA codes for proteins by making RNA, which is going to help turn that code into the protein, and then those proteins are made of amino acids and are going to make traits. So we're remembering DNA, RNA, protein, trait. Also remember, A is going to go to T if we're talking about DNA, and U if we're talking about RNA, and C is going to go to G regardless. So when we begin all of this, I like to think of DNA like this gigantic cookbook. It's got every recipe in the entire world. And each one of those recipes is going to be a gene. In other words, each of those recipes in this gigantic cookbook codes for a certain trait or a certain dish. And how we go from that recipe to that actual finished food, that is going to be this process of protein synthesis. Now, protein synthesis involves two steps. Transcription, where we're making RNA, and translation, where we're making proteins. So when we look at our DNA to RNA to protein, DNA to RNA, that's transcription. RNA to protein, that's translation. I like to think of this as a scribe. When you transcribe something, you are simply making a copy. So going from DNA to RNA, you're just kind of making a copy. When I go from RNA to protein, I am translating it into a completely different language. So I'm going from nucleic acids into proteins or amino acid language. Now we got to look at some of the different types of RNA that we're going to use in this process. We have mRNA, which is what's going to actually carry the DNA code to the ribosome. We have tRNA, which carries the amino acids, remember the building blocks of the proteins, to the ribosome. And then rRNA, which actually helps make up the ribosome. If you remember, the ribosome was made of proteins and is the site of protein synthesis. So if you notice, the ribosome is involved in a lot of things here. It's pretty much, other than the DNA itself, the most important thing involved in this whole process. It's where everything happens. It's like the factory that puts this all together. And so just to help you guys out, M stands for messenger. It's carrying the message of the DNA. T stands for transfer. It transfers or transports the amino acids. And rRNA, the R stands for ribosomal because it helps make up the ribosome. So let's look at specifically the process of transcription. We're going to start out with the code in DNA. And what's going to happen is the DNA code is going to be transcribed into mRNA. And this is all going to be done by enzymes. And remember, we're going from DNA to RNA. So I like to think of it this way, that this huge cookbook with all of these recipes, it's really just too big to lug around. You really don't want it to get messed up. So instead of trying to use it directly to cook with, what you're going to do is you're going to find the recipe you want and you're going to jot it down on a little piece of paper, because who cares what happens to that little piece of paper? You can make another one if you need to, if you spill something on it, whatever, and then you can just throw it away when you're done. And that's basically what your cells are doing. They're taking the DNA and they're converting it into mRNA so that there's no risk of messing up your DNA, and you can just get rid of the mRNA when you're done. So remember, in DNA we have T's, in RNA we have U's. So for example, if in my DNA I have TAC, that's a codon. My RNA, the complementary RNA sequence, is going to be A, because T still goes to A, U, A goes to U, and C goes to G, so I would have A, U, G. So again, if I look at this on a larger scale, I have 4 codons here. After transcription, so if I tell you to transcribe this DNA sequence, you would give me ACU, GGC, UAA, and UGU. So notice you have done you've made a complementary base, and you're using a U instead of a T. It's the exact same thing we've done before, but U's instead of T's. Alright, so in this process, what's going to happen is an enzyme is going to open up the DNA double helix. Because we're only going to use one side as the code. And I kind of think of it like you have to open up the book in order to read it. So then another enzyme is going to come along, and it's going to take these RNA nucleotides that are floating around the nucleus, and it's going to put them together based on complementary base pairing and create our mRNA. So if we start with TAC, it's going to do AUG, and then UUG, CAG, and then UAG. So that's my mRNA, and then it's going to separate, and it's going to go off to the ribosome where translation is going to occur and create the actual protein. And so when we look at this kind of as a drawing, you'll see this in the back of the room. Here we have our DNA, and notice it's been opened up in this one spot to allow for this transcriptional process to occur. We have our enzyme that's doing the job, and we have our mRNA that's being created that can then go off and create the protein.